Over the last 2 decades, genome-wide association studies have identified more than 150 significant genetic risk loci for Alzheimer's and Parkinson's. Yet, despite mapping this vast genetic terrain, clinical predictions remain poor, and single-target therapeutics continue to fail. The problem lies in a pervasive bioinformatics model, the flat list assumption. Current methodologies treat all implicated risk genes as though they act simultaneously, continuously, and within a single tissue compartment throughout a patient's entire life. This approach assigns each locus a distinct odds ratio and aggregates them into a single mathematical sum, known as a polygenic risk score. The result is a static liability number representing a patient's supposed lifetime risk of cognitive decline. This diagram shows the mechanical failure of that model. By flattening dozens of complex genetic variables into a single mathematical equation, we are stripping away the dimension of time. The model ignores the biological reality that genes express themselves in different cellular compartments at entirely different stages of the human lifespan. Treating neurodegenerative genetics as a simultaneous, flat list is mathematically rigorous, but it creates a biologically invalid model of disease. We are looking at a temporally structured architecture. A person's inherited risk is governed by load-bearing variables. Specific genetic alleles remain dormant or clinically silent until the brain reaches a specific biological epoch, where that genetic pathway is forced to bear structural weight. Rotating this data to include a z-axis representing time reveals the architecture. The genetic loci separate into 3 temporal clusters. Phase 1, bioenergetic collapse, ages 20 to 50. Phase 2, microglial collapse, ages 50 to 70. Phase 3, extracellular matrix collapse, age 70 onward. Applying a phase-weighted score identifies the specific decade when a patient's inherited risk becomes structurally load-bearing. Phase 1 operates quietly between the ages of 20 and 50. Anatomically, it is tightly localized to the locus coeruleus and other long axon projection neurons deep within the brainstem. These neurons possess cumulative axonal arbors stretching several meters. Sustaining continuous high-frequency firing across that distance imposes an extreme bioenergetic strain, draining local reserves of the coenzyme NAD+. This persistent depletion triggers the integrated stress response, slowing cellular translation to conserve energy. Under normal conditions, the human brain compensates. But specific phase 1 mutations in genes like NMNAT2 and CD38 compromise the NAD+ salvage network, tightening the bioenergetic constraint until it snaps. As distal NAD+ drops, the NMNAT2 enzyme supply fails, removing the inhibitory block on an executioner protein called SARM1. Once activated, SARM1 rapidly consumes the remaining NAD+, committing the axon to irreversible degeneration. Highly penetrant Mendelian mutations, such as PINK1 and PRKN, act as heavy genetic anchors in this specific window. They drive systemic mitochondrial clinical symptoms register on cognitive tests. Phase 1 is a silent substrate-level collapse, a bioenergetic deficit that steadily preconditions the brain's circuitry for immune catastrophe. Moving into Phase 2, between ages 50 and 70, the biological timeline advances, shifting our focus to the hippocampus and the myelin-producing oligodendrocytes. The primary trigger for this phase is an iron-dependent lipid peroxidation cell death pathway known as ferroptosis. The fatty, iron-rich membranes of oligodendrocytes make them the most vulnerable targets in the brain to this specific chemistry. Genetic defects in the GPX4 defense machinery leave these oligodendrocytes structurally unable to suppress the accumulation of toxic lipid peroxides, leading to mast cell death and myelin debris. The surrounding immune cells are forced to respond. Microglia lose their homeostatic state, drop their protective signaling protocols, and default into a highly inflammatory phenotype known as disease-associated microglia. In this reactivated complement cascade, the inflammatory environment causes complement proteins C1q and C3 to erroneously deposit opsonization tags onto perfectly functional, healthy synapses. Disease-associated microglia, driven by mutated genetic sensors like TREM2 and PLCG2, inappropriately recognize these tags, engage, and phagocytose the working synapses. In phase 2, the brain's immune defenders actively transition into the primary executors of synaptic destruction. Phase 3 covers the terminal biological epoch from age 70 onward. The anatomical focus shifts to the cerebral cortex and the fast-spiking parvalbumin-positive interneurons. These specific interneurons require mechanical and biochemical scaffolding to sustain their extreme firing rates. This support is provided by perineuronal nets, rigid, lattice-like structures made of dense aggrecan proteins that tightly ensheathed the cells. The integrity of these nets relies on a strict molecular balance, a constant tension between matrix-degrading enzymes, primarily MMP9, and their structural inhibitors, known as TIMP3. Inside the parvalbumin interneuron lattice, genetic architecture failure causes MMP9 hyperactivity to drastically outstrip TIMP3 supply. The degradative enzymes overrun the system, digesting the aggrecan lattice and stripping the interneurons of structural scaffolding. Without physical support, the neuron's firing frequency stutters and slows. The consequences for the network are immediate. The loss of parvalbumin scaffolding causes a terminal collapse in the brain's gamma-frequency oscillations, which are required for memory consolidation and cognitive processing. This mechanical matrix collapse causes the widespread network disorganization that clinicians will ultimately diagnose as late-stage cognitive decline. This three-phase structure resolves a 30-year scientific paradox. Why has the exact destructive mechanism of the APOE4 allele, a single amino acid substitution, resisted consensus despite decades of intense study? APOE4 destabilizes the brain across all three temporal phases, deploying a different destructive mechanism in each epoch. In phase 1, APOE4 fails to deliver essential cholesterol, compounding early bioenergetic stress. In phase 2, the mutated protein induces highly inflammatory lipid droplet accumulating microglia. Finally, in phase 3, APOE4 accelerates the complement-mediated attack on perineuronal nets, driving terminal matrix degradation. Patients homozygous for APOE4 face a near-Mendelian, fully penetrant disease state. Their structural damage does not add up, it compounds multiplicatively at every sequential temporal checkpoint. APOE4 is the master variable, precisely because its molecular architecture is perfectly aligned to catalyze human brain collapse at every stage of life. In population genetics, researchers often refer to missing heritability, a gap where mathematical models fail to predict real-world outcomes. But that gap is largely an artifact of using linear models. A flat calculation cannot account for nonlinear epistasis across time, such as an APOE4 lipid mishandling in phase 1, compounding a TREM2 sensing failure a decade later in phase 2. The solution is to replace the flat calculation with the phase-weighted polygenic risk score. Returning to our three-dimensional timeline, we can map specific patient profiles. Look at patient A. Their genome is heavily weighted with NAD+ and mitochondrial mutations. This generates a massive risk spike strictly isolated in the phase 1 window between ages 20 and 50. Now compare patient B. Their genome is weighted with matrix degradation mutations. The timeline remains flat through early and midlife, followed by a massive risk spike in the phase 3 window at age 70 and beyond. For genetic data to offer true clinical utility, a score must identify the exact biological moment when a patient's specific genetic risk will manifest. This three-phase model links drug efficacy to a specific biological epoch. Mistimed interventions are mathematically useless. Administering PARP inhibitors to preserve NAD+ will yield no clinical benefit if the patient is already in phase 3. Similarly, matrix-protecting MMP9 inhibitors offer nothing to a patient undergoing phase 1 bioenergetic stress. This data matrix aligns specific drug classes to the genotype-guided phase-matched intervention strategy. PARP inhibitors are strictly locked into phase 1, TREM2 agonists are locked into phase 2, and MMP9 inhibitors are locked into phase 3. Future clinical trial designs must enrich patient cohorts based on biological age and their phase-weighted risk score, rather than relying on broad, undifferentiated screening protocols. Defeating neurodegenerative disease requires shifting from undifferentiated population screening to a precision medicine that treats the specific genetic architecture exactly when it begins to bear weight.